Hi everyone, a quick update on BioNanomics, a not-for-profit laboratory for the Northeast Indiana community to investigate robotics at every scale. Okay, here's the atomic force microscopy set. Here's a microplate reader, and this is just a video board here for remote meetings where you can have people join the room. And then we have our confocal microscope system here with the Nikon N-Spark detector for fluorescence microscopy. There's the laser stack, and these are the incubators and drug administrators that can both incubate and infuse into the live well plate. Also, this has the MMI cell cut system on the back. That's a laser that when you arm the laser over here, it allows you to cut tissue that's inside the incubator so that you can go in and actually identify single cells and cut them out of the tissue. Yeah, so here is the microscope, and the very first thing we've cut is eye into the tissue. We're very honored that Snyder named its robotics workshop after my company, Medical Informatics Engineering, and that's where they build robots for FIRST Robotics. Snyder visited the lab and I got to show them the microscope and test it out. Snyder.
What? We're gonna show this to Mr. Hisson.
And show this to Mr. Tudor. Tudor would flip.
So Snyder, we're gonna cut that out. Here we go. Watch over here.
Oh. I didn't know if you'd be able to see
this.
the
You'll be able to see it.
go.
There it goes. S and I. It's gonna draw a little circle.
Dude.
Oh, it like cuts them out.
Mm-hmm. And once the tissue's no longer like attached, it falls out of the matrix.
So is it being burned out by the laser?
Burned out by the laser.
Dude.
Yep. So there.
Zoom in as hard.
Yep. So you can't read it. It's too small to be seen by the eye, So this is showing an embryo developing. It's a fly embryo, and I'm just scrubbing here, and you can see, look at that, right at that, around the 60-minute mark, all of a sudden, all the cells move to different positions and take on different roles. And if I go really fast, you'll see like all of a sudden the whole larvae is like segmented and turning into a worm. And like essentially there's a quorum sense. All the cells are talking to each other, and at some point, just like a marching band, they're all on the field. They've actually divided. They've all taken their positions, and then boom, the dance begins, and the cells differentiate and become different things. A couple of weeks ago, I got to visit the Allen Institute in Seattle. Allen Institute was the inspiration for me to form BioNanomics in the first place. The Allen Institute was started by Paul Allen, a co-founder of Microsoft. It's a completely open source lab where science and technology are merged. It focuses on brain research, but other research as well involving cellular development. They've put online terabytes of data that they've gotten from slicing brains in various ways, and you can download all of that information right from the internet. Recently, I've been playing with the Brain Atlas, which is a sequence of every single cell in the human brain and what the transcriptome is within each of the cell types. And it's all freely downloadable from their website. You can see some of the other areas they work on. So BioNanomics is dedicated to inspiring students to get involved in robotics initially, and then into synthetic biology to develop the nanorobotics, which are basically what cells are. The team right now is myself, Bree, Dr. Miro serves on our board. Kim Horner, my wife, is the secretary, and Ashley does marketing. If you're a teacher or a student who's interested in using the equipment to run your own experiments, reach out to us. The new frontier is at the nanoscale, and we can help show it to you.